Bismillahirrahmanirrahim. Now we have the second video on paper 4 to June 23, and we're going to discuss three questions in this video, 5, 6 and 7. Question 5. Warfarin is a poison used to kill rats. Some rats are resistant to warfarin and can survive the effects of the poison. Warfarin resistance is due to a mutant dominant allele at a single gene locus. Researchers investigated the population of brown rats on a large island where there are no predators of rats. On this island, warfarin is used to try to control the rat population. The researchers found that 40% of the population of rats was resistant to warfarin. Use the Hardy-Weinberg equation 1 and equation 2 to calculate the percentage of the population of rats on the island that is heterozygous for warfarin resistance. So 40% of the population of rats was resistant to warfarin. So now you can see the working. You see, the researchers found that 40% of the population was resistant. Now resistance is due to a mutant dominant allele. So this would be p square. P square will be 40% or 0.4. And so we left with Q square. So Q square is 0.6 or 60%. So then of course you find out under root of 0.6, which is 0.775, but the heterozygous, because it says find out the percentage of heterozygous. So heterozygous would be 2pq. So 2 into 0.225 into 0.775 is 35.35, and that's 35%. Now in natural populations, it is usual to find that only some of the conditions of the Hardy-Weinberg principle are met. Suggest and explain which of the conditions of the Hardy-Weinberg principle are most likely to be met for the island population of brown rats. Remember, there is no migration. There is no flow of alleles or genes out of the population or into the population. So no immigration or emigration. Then the population is isolated. Means there is separated by a barrier. And then there is random mating is occurring and rats move around the island. Then there's a very large population of rats, and then rats having a very high reproductive rate, and there are no predators. Then rats are diploid, and they have two different alleles for the gene, so there can be heterozygosity as well. If it wasn't so, then of course this doesn't apply. The Hardy-Weinberg does not apply in such situations. You see, Hardy-Weinberg principle only applies if there were two alleles of the gene, because that is what we use the P and Q for. So P is the dominant, and then Q is the recessive, and then 2pq is the heterozygous. So what you have to understand in such a case, there has to be, number one, no migration. That means no flow out or into the population. Then population is isolated. Then there is random mating. They're not sort of kept in a certain area, and then so that is not very random. If there are only about 100 mice live in a certain area, and then they're not allowed to mix with the other 500. So random mating is occurring. Rats move around the island, and they're ready to go anywhere where they like. They're very large populations, and rats have a higher reproductive rate, and there are no predators. Then rats are diploid, because if they weren't diploid, then we wouldn't have the p square and the q square story, and have two different alleles for the genes. Then coming to part B of the question, dominant advantageous alleles and recessive advantageous alleles both naturally occur in populations. Advantageous alleles, dominant advantageous, and recessive advantageous both naturally occur in the population. Explain why, when a new dominant advantageous allele occurs, its frequency increases more quickly in the population than when a new recessive allele occurs. So you see, the new allele initially is found in heterozygotes. Then the dominant advantageous allele is expressed in this situation as well as in this situation. Selection can act on individuals' dominant allele straight away, while the recessive advantageous allele is not expressed in this situation, not in the heterozygote. It is not expressed because the dominant allele shows its effect in the heterozygous state, while the recessive is only expressed when it is in the homozygous recessive condition. So selection can only act on individuals who are homozygous recessive. Only then selection will occur. It takes time for the homozygous recessive to occur, because you see, If all the parents are this, then this is only a 25% chance of having it. And then if it is this, then there's no chance of it being homozygous recessive. So explain when a new dominant advantageous allele occurs, its frequency increases more quickly in the population than when a new recessive one occurs. So new allele initially found in heterozygotes, then the dominant advantageous allele, we're going to talk about that. So these are the 2 points for that. Dominant expressed in heterozygotes, selection can act on individuals with dominant allele straight away. Then we will talk about the recessive ones. Recessive allele not expressed in heterozygotes, selection can only act on individuals that are homozygous recessive, and it takes time for the homozygous recessive to occur. And coming to question number 6a, figure 6.1 is a photomicrograph of a section through a Bowman capsule and a glomerulus. On figure 6.1, use label lines and letters to label F, the location of the glomerular filtrate. So this would be F. And the location of the glomerular filtrate, and P, the location of the blood plasma. So you could say anything here would be fine. So this would be P. So this is inside the glomerular capillary. So you have to really be labeling it where you can see the glomerular capillaries. Anything in this area would be fine. You can even have a part here. Even here is fine. So anything in this area would be glomerular capillaries would be fine. And the location of the blood plasma, because you see that would be inside the glomerular capillaries. And then we go on to the next part of the question. The glomerular filtration rate, GFR, is the rate at which fluid filters from the blood in the glomerulus into the Bowman's capsule. Figure 6.2 shows the mean GFR values for healthy males and females of different age groups. Now we're going to ask you some sort of mathematical question. Using figure 6.2 to calculate the yearly rate of decrease in mean GFR from the 2029 age group to the 8089 age group for females. Show your working, give your answer to 1 decimal place and include units. There's a lot of marks, 3 marks. So there's 1 mark for the unit, 1 decimal place, and then of course the calculation. So we're going to be talking about 20 to 29 and 80 to 89, and we're going to talk about important things, females. So let's look at the females here. This is the females here, and this is the females here. So let's look at these figures now. In this situation, you can see it is 118 here and 52 here. So 118 minus 52, well, that of course becomes the standard. 118 minus 52. But then what do you divide it by? How many years was it? So you could have done anything. You could have divided it by 60, or you could have divided by 51, or you could have divided it by 69 or 60. So all these are possible. 118 minus 52 over 60, or 118 minus 52 over 69, or 118 minus 52 over 51. So this of course was standard, 118 minus 52. You could have divided by 60, 69 or 51. And of course then the answer would be either 1.1 or 1.0 or 1.3. But that wasn't the situation. The question was, what units are you going to give me? And that was the important thing. So you had to give me the units in centimeter cubed per minute, and you have to give me per year. So you have to give me the answer, which was of course either 1.1 or 1.0 or 1.3, depending on what figures you took. If you say 118 minus 52 over 60, then it was 1.1. Or if you said 118 minus 52 divided by 69, then the answer was 1.0. Or if you said 118 minus 52 divided by 51, then the answer was 1.3. So any of these answers if you had given me, but you must know it. What did it say? Calculate the yearly rate of decrease. So of course you got the figure 118 and 52, but the yearly rate, because this was between 20 and 29 and that was between 80 and 89. So you had to see the yearly. If you said 80 minus 20, 60, even that was okay. 89 minus 29 was also 60. But then if you said 80 minus 29, so all those possible answers could be correct. So please have a look at this very carefully and see if you were able to get this answer correct or not. Then coming to part 2 of the question, the age of a person and whether they are male or female can affect GFR. Suggest and explain other factors that can affect GFR. Well, high blood pressure can affect GFR. Then water potential gradient between the glomerulus and the Bowman capsule can affect GFR. Then kidney disease, like damage to the basement membrane or cancer of the kidney. Then dehydration, lower water potential of blood decreases GFR. Then high altitude decreases GFR. Then you can name some drugs like diuretics or vasodilators. They can affect GFR. Then the largest difference between the afferent arteriole lumen and the efferent causes higher GFR. You see, when the difference between the efferent becomes narrower, then the pressure, back pressure increases. So more blood will be filtered out of the glomerular capillaries. So that would be a higher GFR. What you need to understand is that glomerular filtration rate is dependent upon the pressure. So high blood pressure will higher GFR, means high blood pressure in the entire body system, the aorta, so more in the renal artery, more in the afferent arteriole. This will cause a higher GFR. Water potential gradient between glomerulus and Bowman's capsule. This will affect the GFR. Then kidney disease will decrease the GFR. Because if there is damage and blockage, basement membrane is damaged, then we're going to have problems. Then dehydration will decrease the GFR. Dehydration means less water. Large difference between the afferent and the efferent arteriole lumen. You see, the lumen of the afferent and the efferent arteriole, that is going to affect the, so if it's going to be a very large difference, the efferent arteriole narrows because there is a sphincter around that. If that becomes narrow, then back pressure is created and that's going to result in a higher GFR, which I've just given you in the last point. So any three out of these, there were lots of points. There's also another point for high altitude, but I'm going to leave that out. Then there are drugs also like diuretics, which are going to increase the GFR. ACE inhibitors, vasodilators, but we don't talk about that. I don't think we talk about that a lot in the syllabus, so I'm not going to talk about that. Then of course we come to the part C of the question. The glomerular filtrate passes from the Bowman's capsule to the PCT, where selective reabsorption takes place. Describe the process of selective reabsorption. Very direct question. I mean, I was just teaching it the other day and I said you need to memorize this, and I insist on being very very strict about this, that everybody should memorize it, and you can just keep the mark scheme in front of you, use it as a screensaver for a few days and memorize it. So sodium actively transported out of cells, sodium enters the blood. Sodium concentration in the cells lining the PCT will decrease. Sodium enters the cell from the filtrate by facilitated diffusion. This results in co-transport of glucose. Water potential of the cell decreases, so water enters the cell from the filtrate by osmosis down the water potential gradient. Glucose and amino acids leave the cell by facilitated diffusion. Glucose and amino acids are reabsorbed and moved into the blood, and that of course will get you your 7 marks. That's a lot of marks. 7 marks, and then about 11 10 mark scheme points, and then there's of course any valid point. So everybody should be knowing these points and they should memorize these. I keep on insisting that you all memorize this, and even when I'm teaching you in class. So I hope you all can memorize this. So you can look at the mark schemes here. Sodium actively transported out of the cell. Then sodium enters the blood. This is sodium actively transported out by the sodium potassium pumps, which are on the basal side. So this is the PCT, and these are the cells lining the PCT, which have got the microvilli, and the sodium potassium pump is on this side, and then this is the blood capillary on this side, so that is going to enter the blood. So sodium actively transported out of the cell, then the sodium enters the blood. So the sodium will enter the blood here. Now the sodium concentration in the cells decreases, and then this is the glomerular filtrate flowing in here. So that is going to cause the sodium to move into the cell. And when the sodium concentration in the cell decreases, sodium enters the cells from the filtrate by facilitated diffusion, and this of course results in co-transport of glucose and amino acids. Then when so much of glucose enters the cells, then the water potential of these will decrease. The cells will have so much of glucose and amino acids, so the water potential of the cells will decrease, and water enters the cell by osmosis down the water potential gradient. The glucose and amino acids leave the cell by facilitated diffusion. So all those which are entering the cell, so these are the cells lining the PCT. So the glucose entering the cell will ultimately enter the blood. So the glucose and amino acids moved into the blood, so they will ultimately finally go into the blood, and then we can have any other ADP, other named ions reabsorbed. So there was a whole lot of mark scheme points which you could have given me, and you could have easily got full marks in this question, which is a 7 marks question, which is a very direct question and a very easy question. Then coming to question number 7, a striated muscle cell muscle fiber will contract when stimulated by a motor neuron at a neuromuscular junction. Air temperature can affect the temperature of striated muscle cells. Investigation has shown that the efficiency of contraction of striated muscle cells decreases when the air temperature decreases. Suggest reasons why a reduction in temperature can decrease the efficiency of contraction of striated muscle cells. Now, it's a 4-mark question, and you could have come up with anything. Respiration rate decreases, less ATP produced, reduces activity of enzymes, reduces the movement of calcium ions or sodium ions or acetylcholine or the neurotransmitter. So if there's less acetylcholine broken down because the acetylcholine remains attached to the receptors, or you can say the acetylcholine esterase is inactive. Now what will happen in the sarcomere? There will be less number of calcium ions will bind to troponin, so no cross bridges will be formed, so no power stroke. What will happen in the sarcomere? No detachment of the myosin head, so no cross bridge is broken. So hydrolysis of ATP becomes slower. And of course, all these can result in shivering or reduced blood flow to the muscles, and energy is diverted for thermoregulation. So any 4 marks out of these, and you could have come up with quite a few points. Suggest reasons why a reduction in temperature can decrease the efficiency of contraction of the striated muscles. So reduction in temperature means less respiration rate, less ATP produced, reduced activity of enzymes, and then you can talk of the enzymes that are responsible in muscular contraction. So respiration rate decreases, less ATP produced, reduces activity of enzymes, reduces the movement of calcium ions or sodium ions or acetylcholine. Acetylcholine esterase less active, which means that acetylcholine will not be broken down, so will remain attached to receptors. Then in the sarcomere, pure calcium will bind to troponin, no cross bridges formed, and no power stroke. And in the sarcomere again, no detachment of the myosin head, or you can say hydrolysis of ATP is slower. And so of course, any of these 4 points, and you got your 4 out of 4. Now coming to the B part of the question, the greater blue ringed octopus Neurata produces TTX, tetrodotoxin. TTX is a neurotoxin. If a mammal is bitten by the octopus, the effect of TTX can cause the death of the mammal. Figure 7.1 shows a greater blue-ringed octopus. TTX binds to voltage-gated sodium ion channels to the axon of a neuron and changes the tertiary structure of the channel protein. Binds to the voltage-gated sodium ion channels in the axon of a neuron and changes the tertiary structure of the channel protein. Suggests how TTX may affect the functioning of a motor neuron. Well, of course, the sodium channels don't open. When the sodium channels don't open, or they will not open, or they will stay open, then the axon membrane cannot be depolarized, or it will stay depolarized. So no action potentials will be generated, or continuous action potentials will be generated. Calcium does not enter the presynaptic knob. So if calcium does not enter the presynaptic knob, vessels will not be stimulated to move, so no acetylcholine will be released. Or we can say calcium always enters the presynaptic knob and the vesicles will always be stimulated, so acetylcholine will be continuously released. And this, of course, would result in paralysis, or it would result in continued muscle contraction. You see, there are two ways that you could handle this question. Either you say sodium channels don't open, or you say they stay open. So let's talk about the first one now. Let's not talk about the second one, because this is going to confuse you. I know that. So let's talk about sodium channels do not open. So if the sodium channels do not open, then the axon membrane cannot be depolarized. So no action potential is generated. So calcium does not enter the presynaptic knob. So this is going to result in paralysis. How will the muscle work? Muscles will not work. Muscles are paralyzed. If you say the other way around, let's look at the other way around. The sodium channels stay open, so the axon membrane stays depolarized. And this results in a continuous action potential going along the motor neuron. So calcium always enters the presynaptic knob and acetylcholine is continuously released. This will result in a sustained or a continued muscle contraction. And you see, if the muscles contract and they don't relax, you see, when you're breathing, muscles contract and relax. In all situations, muscles contract and relax. When you move your arms, muscles contract and relax. Sphincter muscles contract and relax. When you're breathing, intercostal muscles contract and relax. So all that will result in, it says, how TTS may affect the functioning of a motor neuron. And then it's told you in the question as well that if a mammal is bitten by the opticus, the effect of TTX can cause the death of the mammal. So that has also been given in the question. So you can handle this question very effectively in either of the two manners which I have just explained to you. So sodium channels do not open, axon membrane cannot be depolarized, no action potential generated, calcium ions do not enter the presynaptic knob, so no impulse in the motor neuron, so muscles will not contract, and this results in paralysis. This is one answer. This is one possible answer, and of course the other one which I have just told you, you can have a look at the mark scheme and see the other possible answer as well, but this will get you your 3 out of 3.